Welcome back to another video. In this video we will learn about DNA, blueprint of life. Blueprint of life, DNA, or deoxyribonucleic acid, is the molecule that carries the genetic instructions for the development, functioning, growth, and reproduction of all known living organisms. DNA has a unique double helix structure, resembling a twisted ladder or spiral staircase. This iconic structure was first discovered by James Watson and Francis Crick in 1953, based on the work of Rosalind Franklin and Maurice Wilkins. Base pairing: DNA consists of two long chains of nucleotides that are complementary and held together by hydrogen bonds between specific nitrogenous bases. The four nitrogenous bases are adenine, A, thymine, T, cytosine, C, and guanine, G, which pair up in a specific manner, A with T and C with G. Genetic code: the sequence of nucleotides in DNA forms the genetic code, which determines the unique characteristics and traits of an organism. The order of the bases along the DNA molecule encodes the instructions for building proteins, the essential molecules responsible for the structure and function of cells. Genetic diversity: each individual has a unique DNA sequence, except for identical twins, who share the same genetic makeup. This genetic diversity results from variations in the arrangement and combination of nucleotides within the DNA molecule. Genetic inheritance: DNA is passed down from one generation to the next through the process of inheritance. Offspring inherit half of their DNA from each parent, resulting in a combination of genetic traits from both maternal and paternal lineages. DNA replication: before cells divide, DNA must replicate to ensure that each daughter cell receives a complete set of genetic information. During DNA replication, the double helix unwinds, and each strand serves as a template for the synthesis of a new complementary strand, resulting in two identical DNA molecules. Mutations are changes in the DNA sequence that can occur spontaneously or as a result of environmental factors such as radiation, chemicals, or errors during DNA replication. Mutations can be beneficial, harmful, or neutral, and they are the raw material for evolutionary change. DNA packaging: the human genome contains approximately 3 billion base pairs of DNA, which must be tightly packaged to fit within the nucleus of a cell. DNA is wrapped around histone proteins and organized into structures called chromosomes, which become visible during cell division. DNA analysis is a powerful tool used in forensic science to identify individuals, determine biological relationships, and solve crimes. DNA fingerprints, based on unique patterns of DNA sequences, can be compared to databases to identify suspects or exonerate the innocent. Scientists can extract and analyze DNA from ancient specimens such as fossils, mummies, and archaeological artifacts to study the genetic history of extinct species, ancient human populations, and evolutionary relationships. Researchers are exploring the potential of DNA as a computing material due to its massive storage capacity and parallel processing capabilities. DNA computers could revolutionize fields such as data storage, cryptography, and bioinformatics. Epigenetics refers to changes in gene expression that are not caused by alterations in the DNA sequence itself, but by modifications to the structure of DNA or associated proteins. Epigenetic changes can influence gene activity, development, and disease susceptibility. In addition to nuclear DNA, humans and most other eukaryotic organisms also possess mitochondrial DNA, mtDNA, which is inherited exclusively from the mother. Mitochondrial DNA is essential for energy production within cells and is used in studies of maternal ancestry and population genetics. Cells have sophisticated mechanisms for repairing damaged DNA to maintain genomic integrity and prevent mutations. DNA repair enzymes can correct errors that occur during DNA replication or repair damage caused by environmental factors.